Okay, good morning. Today we're going to talk about imprinting in a brief and fairly cursory way. The idea is that you should understand that this phenomenon exists and something about how it originates. Okay, so the imprinting is the basis for a form of monoallelic expression. Okay, now monoallelic expression means that even though we've got two alleles of each gene, only one of them is expressed. So the one that we're familiar with, of course, is X chromosome, where one chromosome is silenced in mammalian females, leading to the fact that you have essentially monoallelic expression of genes on the X in each cell. Okay, so the idea is, of course, we have two alleles, and that for most genes, both of them are active. But there's some very important exceptions to this rule, where either dad's allele is expressed or mom's, which means that we have only one functional copy of that particular gene. So this is a rare thing. There's only a few hundred total genes in humans that are imprinted, but it's very important. Okay, so it's a process whereby one or the other is shut off, but then it will have to be reactivated later during gametogenesis. Okay, so the motivating thing here is that a long time ago people were doing pronuclear transfer experiments, during which we learned that a zygote needs to have a paternal genome and a maternal one. Okay, so your standard mating of one sperm and one egg gives you one nucleus from dad and one nucleus from mom, and that gives rise, of course, to a normal embryo. But if you do pronuclear transfer and make a fertilized egg, which has in fact two maternal nuclei or two paternal nuclei, you find that embryonic development fails, that the cell or the new embryo needs to have contributions from both parents in order to undergo normal embryonic development. Okay, so that's illustrated again here, that to get normal development, you need alleles from both parents. Well, it turns out you don't need the entire paternal genome or the entire maternal genome. It's the selective activity of these monoallelic imprinted genes that determines what genes are important. Okay, so much work, much work, amazing work, determined that some genes are silenced in one of the genomes and vice versa. And so the result is the maternal and paternal genomes are not equivalent in expression during embryonic mice or people, and the process is known as imprinting. So the idea is one allele is expressed and the other is silenced from each parent. Okay, so here's kind of a representation of what's going on. Okay, in the maternal allele, we're talking about a region of the genome that includes the insulin-like growth factor 2, and it also encodes a non-coding RNA. And there is one enhancer and something called the insulator, which is now called the imprinting control region. Okay, so this situation is going to lead to an asymmetric expression of the H19 non-coding RNA and the insulin-like growth factor from the maternal or the paternal allele. Okay, so in this particular setting, the maternal allele is bound by the insulator is bound by the protein CTCF. It binds the control region, and as a result, we express the H19 long non-coding RNA. The H19 RNA is located adjacent to an enhancer, and the binding of CTCF guarantees that it's H19 that's actually expressed. Conversely, on the paternal allele, the CTCF binding site is methylated, and that prevents the CTCF protein from binding. As a result of that, the enhancer activates the insulin-like growth factor instead of the non-coding RNA, leading to its expression. So we see the asymmetry here. Mom's allele does not express the insulin-like growth factor, but dad's allele does. So in the early embryo, we will see expression of the insulin-like growth factor, but only from one allele, and that's the one that comes from dad. Okay, so one important molecule here is 5-methylcytosine. So this actually establishes a silencing mark in which the normal cytosine is modified to form 5-methylcytosine. Okay, so here's a representation of what we just said before about CTCF binding and insulin-like growth factor expression from the maternal versus paternal allele. So it's showing the same thing with a slightly different cartoon, in which we have, of course, H19 expressed from the maternal and IGF-2 expressed from the paternal. Okay, so the converse situation is seen with another imprinted region, which encodes the insulin-like growth factor receptor, IGF-2R. Okay, in that case, the receptor is expressed from the maternal allele, not the paternal allele, and the paternal allele instead expresses a non-coding RNA, and that is basically dictated by the state of methylation of the promoter of the non-coding RNA. Okay, here's a slightly different representation of that same pair of genomic regions where you see the CTCF binding, etc. And I think it's a little clearer on the nature of the long non-coding RNA air from the paternal allele and the expression of the insulin-like growth factor receptor from the maternal allele. So in this case, the methylation actually prevents expression of the long non-coding RNA and allows expression of the insulin-like growth factor receptor. So we have a reciprocal situation here where dad's allele is expressing the insulin-like growth factor and mom's allele is expressing the receptor, but not both. Okay, so sometimes this situation can lead to disease because after all, we only have one chance to get it right. You can get a good allele from one parent, but if that's the silenced allele, that is going to cause problems. Okay, so we have to look at this cycle of imprinting erasure during embryogenesis, well, during your life and during embryogenesis, in order to see how this is going to work. Okay, so starting where? If we start with the zygote, we're showing imprinted gene number 1 and imprinted gene number 2, where one is expressed from the maternal allele and one's expressed from the paternal. So those imprints are maintained during the life of the individual in your somatic cells. You've got one active allele, one from mom, and one active allele from dad. But those imprints need to be erased in order to establish the next generation of imprints. And it is in the germ cell line that those imprints are erased and then reestablished according to the sex of the individual. So irrespective of which allele you got from your mom or your dad, you're going to erase those imprints and make your own maternal or paternal allele depending on your sex. So once that's been erased and then reestablished, when we form zygotes, we will have imprints which are characteristic of our own gender. So all of our kids will get our maternal one or our paternal one, depending on our sex. Okay, so in somatic cells, mom's and dad's imprints are maintained. Your gender doesn't matter, but old imprints erased and the new one established. And so, of course, in females, we will silence the insulin-like growth factor 2 gene, permitting expression of H19. Males conversely express that growth factor and silence it, etc. Okay, so the poster child for imprinting diseases, poster children, I suppose, are Angelman's and Prader-Willi syndrome. Okay, these are two parent of origin diseases, and they result from nearby mutations within the same genetic region. Okay, this particular imprinted region is a little bit different from some of them in that the neurons have a different pattern of expression for most cells. Okay, and it's the mutations in the Prader-Willi syndrome lead to Prader-Willi, mutations in a region called AS and Angelman syndrome. Okay, and there are actually deletions that span that region, and that will give diseases according to the parent of origin. We're going to consider Angelman syndrome primarily. Okay, here's the Prader-Willi Angelman syndrome region. The blue genes are expressed from the paternal allele, and the red ones are expressed from the maternal allele. Okay, the other genes in this region are not actually imprinted. Okay, so saying that there's a difference in neurons versus non-neurons, which is odd. In non-neurons, the UBE3, which is the Angelman syndrome gene, is actually expressed from both alleles, whereas the Prader-Willi syndrome gene is not. It's only expressed from the paternal allele. Okay, in neurons, however, there is an asymmetry where the UBE3 gene is expressed from the maternal allele, but the paternal one is silenced. Now that leads to an asymmetry in when you have children, when these people have children, who is going to suffer from defects of mutations in UBE3, and either they will suffer or not, depending on which parent gave them the mutant allele, remembering that only one is active in any individual. Okay, so here's a pedigree showing Angelman syndrome, and this takes a little getting used to. Okay, they did us a favor by putting little dots in the people that are actual carriers of the UBE3 mutation. Okay, so remember this is expressed from mom's allele and not from dad's. Okay, so remember the squares are males and the circles are females. Anybody with a little dot is a carrier, and anybody with a filled-in figure is a person with the disease. So this kind of shows you how this all works. Okay, so in the left part of the pedigree, we've got Grandpa, and he is a carrier, and he passes that mutation down to half of his kids, because remember, he's got a good allele, but it doesn't matter in terms of their phenotypes, because they got the mutant allele from their dad. It doesn't count. Mom is normal, right? So he's got a daughter who is a carrier, and she got her mutant allele from her dad, so her allele is not being expressed because it's the paternal one. She gets a good allele from her mom, and therefore is normal phenotypically. But she did get a mutant allele from her dad, which she can then pass down to her kids. Okay, so the father of the children isn't shown here, but we can see that she has two children, both of which have Angelman syndrome, one boy and one girl, because mom really had a 50-50 chance of passing down the bad allele to either child. So the wild-type children aren't shown here. Both kids got the mutant allele from their mom, and they are showing the disease because even though they got a good allele from dad, this doesn't count. Okay, the other side of the pedigree shows similar things. Grandpa is a carrier. He passes the mutant allele down to half of his kids. Well, we've got one girl that's a carrier. This girl has kids, and one of her children, a male, didn't get the mutant allele because after all, it's only half and half. But her daughter does have the mutant allele, which she got from mom. So the fact that both of those children got normal alleles from their dad doesn't protect the daughter from having the disease because dad's allele is silenced. Is this weird or what? Okay, so these slides basically just say what I just said. Who inherited the disease from their mom, etc. Okay, let's just step through this. You can all read this. Okay, so Prader-Willi is kind of the opposite and a little more complicated, so I'm not going to really say much about it, except to say that it's caused by mutations in the same region as the mutations that cause Angelman syndrome. But the imprinting region is what distinguishes these two. Okay. Again, we have expression in neurons and non-neurons, but in the case of the Prader-Willi, it's actually expressed, it's silenced appropriately, it's silenced in the paternal allele in both types of cells. Okay, and the region involved is really quite large, involving numerous genes. And in the human population, a lot of the cases arise from actually large deletions encompassing most, if not all, of this entire region, including the Angelman syndrome region as well. Okay, so here's what it looks like. Many, many genes are encompassed. There are a lot of non-imprinted regions in the same region, but depending on whether you inherit from mom or dad and the extent of the deletion, you will get either Angelman's or Prader-Willi, depending on that. It's a very strange thing.